Alright, so in this video I'll be going over the parasitology cycles, life cycles. So as you know, INI likes to ask quite a bit of these cycles. So what they will usually do is just give you an image of the cycle and then ask a follow-up question. So for example, identify the organism, or maybe they can ask you the drug of choice for that organism, or some you have to find the correct or incorrect statement about that organism, or it could be anything like that. But for all of that, you first need to be able to identify the organism from the given life cycle. So that's what we will be doing in this video. I will be going through all of these life cycles and just highlighting the important points which you need to be able to identify the organism. Obviously you don't need to remember the entire life cycle, and as far as I know, INI always uses these CDC images, standard CDC images in the exams. So I've just compiled all of those CDC images in this PDF. And before we begin, another thing, you also need to be thorough with these three things: which parasites cause skin penetration, which parasites cause autoinfection, and which are not non-biological. This one is not so much important for the life cycle purposes, but these two are definitely important, because from the life cycle you can make out if the organism is being transmitted by skin penetration or autoinfection, which can help you narrow down the diagnosis. Okay, so with that out of the way, let's begin. So the first is amoebiasis. So for this, the you have to just look for this thing. You see, as you know, in amoebiasis, when these cysts enter the intestine, it can be like they can just stay inside the intestine, and so that will be non-invasive colonization. They can actually cause dysentery, so that will be intestinal disease, or they can go to other parts of the body, for example amoebic liver abscess. So you have to look out for this thing, three different outcomes, and it will be also shown in this diagram here. Okay, next is Naegleria fowleri. So for this you have to look out for this nose word penetrating through the nasal mucosa, water entering into the nose, and this PAM, primary amoebic meningoencephalitis. Although this just straight away gives you the diagnosis, so I don't think this is very likely to be asked in the exam. Next is Balantidium coli. I couldn't really find any notable points in the life cycle, and it has never really been asked either. Just the one thing that they asked is this image of the B. coli cyst, which if you have solved the PYQs then you might be knowing, but the life cycle as such is not really that notable or important in my opinion. Next is acanthamoeba. So for this you have to look out this three entry routes. It can be entered through the body, through the eye, through the nasal passages, or through ulcerated or broken skin. That's about it. Next we have Giardia. So if they give you the cycle of Giardia, you should be able to see this trophozoid form, very classical image. And another important point about it is that the trophozoids are passed out into the stool, but they do not survive into the environment. It's just the cysts that survive and continue the life cycle. Next we have Trichomonas. This is very simple. So you can see it is transmitted by sexual intercourse, and there is no cyst stage in Trichomonas. Next we have Leishmania. You just have to look out for this sandfly thing. Out of all the parasites in this PDF, this is the only one which is transmitted by a sandfly. So just look out for the vector, how the Leishmania is entering into the body, so which is through the sandfly. Next we have African trypanosomiasis. Again, this is the same as Leishmania. You just have to look out for the vector, which is the tsetse fly in this case. Then we have So that was Trypanosoma brucei. This is Trypanosoma cruzi. Again, just search for the vector. Try to mean bug. Alright. Next we have Cryptosporidium. So for Cryptosporidium, one thing is that you can see there's the auto-infection. It is transmitted by auto-infection, and the other thing is you will be able to see two kinds of cysts here, two types of oocysts, which is thick wall cysts and the thin wall cysts. So yeah, that's what you have to look out for. The thin wall cysts are what is involved in the auto-infection, and the thick wall cysts will go into the environment and continue the life cycle. Next we have Cyclospora. So for Cyclospora, you have to look out for this part. The oocysts that are excreted in the feces, they are unsporulated, and they are shed as unsporulated, but they need to spend some time in the environment until they sporulate and become infective. So this exact point is also one of the PYQs which has been asked, so it will be helpful to remember this. Next we have Cystosoma. So we come to the trematodes now. So as you will be knowing about the trematodes, you have to look out for the vector. So the primary intermediate, sorry, not the vector, the intermediates. The primary intermediate for all of the trematodes is the snail. So if you see a snail in the life cycle, you already know that it's a trematode. Then you have to look for how the organism is entering into the body. So for all of the trematodes, the infective part is the metacersaria. As you can see here, this is metacersaria, except for one, which is Cystosoma, for which it is cercaria. So if it's cercaria, then you automatically don't know that it's a Cystosoma. And you will also be able to see these different parts which are infected by the different species. For example, the Cystosoma hematobium, of course they won't mention the names here, but yeah, Cystosoma hematobium infects the bladder and so on. Then we have Clonorchis. So as you can again see, we can see the snail, which is the primary intermediate, and if the secondary intermediate in Clonorchis is fish. So if you see this combination, then it is Clonorchis. Then we have Fasciola. So another trematode. As you can see, the snail primary intermediate, and here the secondary intermediate is fresh aquatic vegetation. And as you can see, the infective part is the metacersaria. As I said, for all the trematodes it is metacersaria except Cystosoma, for which it is cercaria. Next we have Diphyllobothrium latum. So here we have to look out for two intermediates, the fish and the cyclops. Cyclops, or as they have called it here, crustaceans. Next we have Taenia. Taenia is pretty simple. You can see the pig and the cattle. So for Taenia solium and Taenia saginatum. Echinococcus, again very easy. You can see the definitive host, which is the dog, and the intermediate host, which is the sheep. Next we have H. nana. So here you will be able to see a separate cycle for auto-infection. I hope you remember the organisms, the parasites which are transmitted by auto-infection. And another notable point is that H. nana doesn't have any intermediate host, as can be seen from the cycle. In this case, this rat isn't the intermediate host, but just like humans, it is the definitive host. Next we have Ascaris. For Ascaris, the most important part is this one, that the larvae enter the circulation and go to the lungs, where they can cause Loeffler syndrome. So you just have to look out for the migration to the lungs. If you see that, then that's Ascaris. Next we have intestinal hookworm. So for this, you just have to look out for this image of the feet, because they can infect the body by penetrating the skin. There are two other parasites, which is Cystosoma and I'm forgetting which one, but they can also penetrate the skin, but the image of this foot is only given in this life cycle for the hookworm. Next is strongyloids. Strongyloids, again you have to look for auto-infection, and other than that I couldn't really find any notable points specific to strongyloids, but you'll be able to see these two cycles, free living and parasitic. Maybe you can remember it by that. Next is trichinella. Here we have the two separate cycles, domestic cycle and sylvatic cycle. Next, E. vermicularis. E. vermicularis, the classic point associated with it is that it causes perianal itching at night because the female migrates to the perianal region at night to lay the eggs. So just look out for this thing. Next we have Wucheria bancrofti and Brugia malai, which cause filariasis. So these two cycles are pretty much exactly the same, as you can see. To identify filariasis, just look out for this mosquito as a vector, and they infect the lymphatics. You can't really differentiate between Wucheria and Brugia as such from the life cycle, but I don't think they will ask that in the exam. Next is Loa Loa, so just look out for Chrysops as the vector here. Onchocerca, similarly, black fly is the vector, just look out for that. Babesia, again, tick is the vector, hard tick or Ixodes tick. So yeah, just look for that one. And last is Isospora. Isospora, you'll be able to see these two different cycles, asexual cycle and sexual cycle. So you can identify it with that. And I think that's about it.